No, the one that goes bing, bing.
Did you get 20? I got 25. I got 25. That's your GPA. Don't find it. 25.
No, I like that one.
That's still a lot.
Guy.
Oh, sorry.
All right. So, shh, quiet.
Yeah, you're good.
Can you face it more towards this way a little bit? Don't move it too fast. Right, move it up. Right. So. Oh shit. Hey Frank, can you do a quick favor here? I said that bring the ball.
Is that what? I should give you a timeline grade. I'm not grading any.
I'm reading timeline. Are you going to grade them?
Alright, number one. Sorry about that. Okay. So, we're going to try to answer these questions together. And as we do so, maybe hopefully kind of review this topic. Alright, so what are the functions of the cell membrane? Marjorie, give me one thing the cell membrane is for.
It controls what comes in and out.
Controls what comes in. Oh, pause actually. Before we actually talk about the cell membrane, I wanted to clarify what you have to do for this review project. Okay? Now, 6 weeks ago, actually now it's 7 weeks ago I assigned the timeline. Right? And then 1 week before it was due, people started freaking out about how to do a timeline and stuff like that. Alright? It was too late to do a good job at that point. It really was. Now, this one, the timeline was hard because you have to unscramble things, get pictures for everything, and then there was a lot of busy work, a lot of things to write down and stuff. This is easy, but it's a lot of work. If you leave it for the last minute, you literally cannot do this 3 days before it's due. It is literally impossible, all right? It's too time consuming. It takes about 30 minutes per lesson if you do it right. And there's like a lot of lessons, right? So this is like a good 400 to 500 minute project. So if you do it could take 10 hours to finish it. All right? Now, it's not the kind of thing you're going to do last minute, okay? And if you do it last minute, it's going to defeat the entire purpose of it. Let me explain what you need to do, right? So, by the way, before I talk about this, archive it again. Sorry about this. I don't like to do this, but in order. First, next class. So the cell that we're going to build is the size of one of these tables. All right? Which means if you have the membrane, or the wall, or the plasma, or cytoplasm, you're not doing it alone. You're doing it with someone else. So whatever you're doing, you got to agree and talk about what you're going to do, and do it as a team. For example, if you have a membrane, and you have 4 people, you split the perimeter in 4, and you're going to do that perimeter, but everybody has to do the same thing, and it's got to look right. So when I look at your Golgi apparatus, it's got to look like a Golgi apparatus. In addition to that, you have to prepare a 30-second speech where you're basically saying why I should vote for your organelle or your structure, because not all of them are organelles, as your structure as the most crucial. Even if you think that it's not the most crucial, you should still try to convince people to vote for it. The idea is, by the end of that, we're going to know what everything does and how it looks like. Yes? Everybody in the group has to say something. Right? So, in fact, I'm not even going to go in order. I'm just going to go around. And so it's not going to be like, so we're going to just, even if you said the same thing, it doesn't matter. But you should practice what you're going to say separate from them. That's not a group. It's not actually a group assignment. It's just that since you're building it together, you got to build your own piece, so to speak. Okay. The other thing, now the project. So there's 2 parts. The first is a worksheet. You have to download the worksheet. Don't try to access it. It won't work. You got to download it. Link on the top, download. Once you download it, one has the answers, the other one has the questions. All the answers are there. So it's like that one, if you don't do it, seriously, if you don't do anything, you're going to get a 0, like a 0. I don't like giving zeros. I give like 50s because on the 100% scale, the zeros really hurt. I will literally give a 0 if you don't do anything because the worksheet has the answers posted, so you could copy the answers, right? So you're not supposed to do that, but if you don't, you can't even do that, then it deserves a 0, right? Now, let me explain what you're supposed to, obviously I don't want you to copy the answers. Here's what you need to do. If I were you, this is what I would do. I would open the worksheet first, and I would try to do it with no help. Just go through it and see if I can do it. All right? You do not have to print it, but if you don't print it, you have to copy all the questions. So you're printing it, right? Now, if you don't printing it, if you don't print it, you can copy all the questions. Now, you have to have all of it done, okay? All the pages assigned done. However, the first time I look at it, I'm going to just try to do it without any help. Don't even look at the answers. That will tell you right there what you're the weakest in, what topics that you know the least about, and it will also tell you how much biology you know by heart. Although it's different to know biology, here's the thing. The EOC is not just factual, it's application. Some of those questions in the worksheet are application, but a lot of the workbook is also application. By the way, those who are wanting to do really good on the EOC, remember that the Biozone lessons are a lot of application. I like them a lot for the purpose of that. And remember that now you also have another type of worksheet that you can do on lab days that are open. I know we haven't had one of those in a while, but we're going to have them again where you guys choose what to do. And one of the things you can do also is the new type of assignment that I posted is the same place, textbook resources, where the Biozone are. There's a new one there that you guys can do, which is it's called POGIL, P-O-G-I-L, right? It's really good worksheets. They're like high-level conceptual thinking, and those are still worth 1 point per question, while regular worksheets are 1 point for every 2 questions, right? So if you are doing worksheets, do POGIL worksheets instead of the factual-based worksheets. Are you with me? Because they're going to challenge you to think more, and that's better, okay? These are going to be a little more like that. Some of those questions are less factual. Now, what you need to do is that after you're done with the worksheet, you tried it, went through it once, then you start working on this. The first thing you do is the baseline assessment. So you take the baseline assessment. I suggest what you do is you literally rip it out of the book, all right? Make it look nice, hole punch it, put it in the binder. Answer directly there, all right? And make sure you put a cover page that says, baseline assessment. Then you put a cover page that says Unit 1, Chapter 1, whatever, and you do this. And you do this for every chapter. This is what you do. Every paragraph. And it's like, if it skips a line, it's a new paragraph. So let's say I'm doing molecular biology, that's what we're doing right now, right? So that's a paragraph, that's another, that's another, that's another, that's another. Every time it skips, it's a paragraph. Every paragraph, you just read it, tell me what it was about. That's it. Okay? Some of them are super simple, some of them are harder. Then, every bolded word, every key term has to be defined. So first, do all the main ideas. You're supposed to number them. Main idea number 1, 2, paragraph 3, paragraph 4, paragraph. Are you with me? When you finish all the main ideas, you define all the key terms. When you define all the key terms, you copy all the charts and tables. You finish all the charts and tables. At the end of each chapter, there is a questions. So you rip the questions out, answer them, put it in the packet. Hole punch it, put everything in a binder with a cover page. Do the next chapter. Every chapter will take you 30 minutes to do it if you do it right. Now, there's a lot of chapters. Like, seriously, there's 10. Right? So if it takes 30 minutes per chapter, there's a 300-hour assignment. 300-minute assignment. 300-minute assignment is 5 hours. And that's, however, you don't do every chapter right now because we're skipping a few because we haven't covered them yet. Right? So you're only the ones which says to do it right now. Later in the year, we're going to do the rest of them. But for now, just the ones which are for the midterm. Is that very clear? Now, in addition to that, now I'm done. Did the workbook chapters. Save the workbook for later in the year. Then you're going to go back to the worksheet. Now you try to do the ones you couldn't do before. See if you can do them now. If you still can't do them, then look up the answers and copy from the answers. You follow what I'm trying to say? That way you practice everything. Then when you see the 1 week before the midterm, you review, especially the parts that you were the worst on, right? That you really knew the least about, and then hopefully kill the midterm. Also, remember how I did that data thing for the test, for the ecology? By the end of this week, I'll have the evolution one done as well. So you'll be able to see which objectives you sucked at. Right? You'll also be able to see the ones you were good at. You're also going to have that same information for the cytology test, which happens on the week we come back from the break. Right? So, and that will count for the 39 weeks. The timeline, we're not sure yet, but the next test will count. And that test, that will count for the second 9 weeks, right? I will also have that data posted before the midterm happens. So that way you will be able to tell which objectives you should hit right before the midterm. The 9 before, you should hit those objectives, the weak ones. Are you with me? Because you're really only going to be graded on the ones that, well, you're going to get the midterm, which gets sent out to the state, whatever. But for my class, your grade is going to come from the ones that you should have known. Is that clear? So because you're going to get questions about, it's kind of like taking another baseline. So it's not going to be the same test, but it's going to be a similar test. And in another test, it's some of the questions will be will be about the same material as there was in the baseline. But some of the questions will be stuff that you haven't covered yet. But because you've learned a bunch of stuff, in fact, 70% of the content has already been covered by the time we take the midterm, right? That means that you should do a lot better than you did in the baseline, especially on the stuff that we covered, correct? Now, this assignment, you should have started already on this assignment. The day that you've turned in the timeline, this became the new thing to worry about, right? Now, you cannot do the same thing you did for the timeline because although it's not hard, it actually isn't hard. It's just like busy work. But don't treat it as busy work. Treat it as study work. Are you with me? Review this way and make sure that on the midterm you know all this stuff. So if there was, by the way, this is EOC stuff. I've been teaching you more than just the EOC, right? But this will review what the EOC stuff is. A good, honest student can do this in 2 weeks and pass the test with a 5. That's why I'm telling you, this is not what the class is about. The class is about more than that. Now, here's the thing, and I repeat this because I know I asked you, a lot of you guys. But here's the thing. When I teach you on my videos, those of you who are watching my videos have been learning, have been exposed to a lot of biology. But my videos in the advanced notes are up here. On the quiz, I asked you down here, right? The EOC just wants this. Understand? So my standards of what I show you is really high. But what I actually require of you is not that high. I'll give you an example. The timeline, yes, it was 150 facts. But if you had done, there was one person in the 5th period who did 80 mistakes. Now there's 300 points because there's how many facts was it actually? 142. All right.
Wasn't it 43?
100 something points. All right, whatever. Double those is how many points you could have gotten. So you could have gotten 250 points about. Actually, 300 points about. All right, but you could lose those points depending on two things. Is it out of place or is it out of period? Right? But then you get some bonus points for creativity. But you also lose points if you're missing all the fats, if you're missing the color coding, if you're missing the pictures, if you're missing the titles of the periods, if you're missing the dates for the periods. So all of it together, you get 50 points of creativity bonus and out to 50 minus points if you're missing certain things. And then you start with 300 points because it's like more than, actually I'll give you like 10 points for free. You start with 300 points and then you lose points depending on what you don't have. But those of you who have graded, and those of you who haven't, by the way, you should grade it. Because that's the best way to find out what the real order was supposed to be, so you learn the right order. So you graded somebody else's, and by the way, when I give yours back, check the rubric against yours and see, okay, I get it. This is why I did wrong. All right? So because you don't want to go out there knowing the history in the wrong order. So you want to make sure you look at the right order. So you should grade at least one of them, right? Every one you grade gives you 2%. By the way, there are only 47 turned in, right? I should know more. 50 turned in or so. If one person grades, if everybody that comes to grade grades 5, quickly they will run out and you won't have the opportunity anymore, right? But everybody that grades it gets 2% more for that timeline. If that never happens, I'm grading 1 or 2 per day. And at this rate, at some point in the 39 weeks, you'll have the grade back. Are you with me? But if you grade it now, it will go in now instead of the 39 weeks. All right? So it's up to you if you want to do it or not. Now, it takes about 15 to 20 minutes when you know what you're doing to grade each one of them. But when you do grade them, you notice that I was actually very lenient. You could be actually, the fat could be out of place and still be okay. Right? So with all of that, and then on top of that, it works like this. I still curved it on top of that. So that means for you to do bad, Yes, I asked for a lot, and it was material that was not even EOC. But when you actually think about how I grade it, it's down here. You feel what I'm saying? However, it is worth half a test grade. So those of you who haven't done it, I mean, that's going to cause brutal pain to your grade. It will drop you like a couple letter grades if you haven't done it. All right? Now, seriously. So that's a problem. So you've got to do it, even if it's late. Got to get that done. All right, let's talk about this really quick.
I have a question.
Yes, questions.
Okay. For that, is everything handwritten for the work?
Except for the questions, which you can rip it out and answer it directly there. Right? But everything else is handwritten. Yes. Hi. I can't do it now because I'm teaching. I have to stop to do it. So, all right, how's best for you? You never went?
What?
You never went to
No.
You're crazy. All right, so,
Question 2.
yes.
So, okay, you want everything in a binder?
Yes, organized in a binder. Yes.
Handwritten? Okay.
It won't actually be too thick of a binder. Like those half-inch binders should be enough.
Really?
Yes.
Do you have to copy the paragraph?
No. I never said copy the paragraph. What did I say?
Main idea.
Main idea. So if the paragraph is this thick, you just have to write one sentence about what it was about.
Oh, it's one sentence? Oh.
Main idea. So, right? Yes.
Okay, so on the website you have like what pages, like what page to what.
Everything is clear on the website. All right. It's already been there. The fact you're asking me that question is a bit scary though. Okay.
Oh, yeah. Every single
Sad
picture.
face. All right. Now listen. All right. So Melanie,
Not what?
Melanie, give me something else. Okay, we already talked about cell membrane as something that controls what comes in or comes out, like the customs department. It's something that contains the volume of the cell, the organelles, and the cytoplasm. By the way, cytoplasm is the region. The liquid is called cytosol. We talked about this last class, right? What else the cell membrane does, Melanie? Give me one more.
Allow cell
Cell recognition, right? There's proteins. What else? Somebody else knows. Yes, Frank. Yeah, the shape of the cell is part of it. Yes. What else? Watch out. If you say something, there's a Lego game throwing a Lego at you. What else? Anybody? Open forum. Yes. For things to connect, yes, to each other. Cells to connect to each other. Or even the cytoskeleton to connect to. All right? So there's a lot of functions. Some things even move with the help of the cell membrane. Check this out.
New white blood cell.
This is actually pretty cool. Check it out. Shh, quiet. So that is a white blood cell attacking a bacteria. It's going to try to attack a bacteria.
Hi, this is Dr. O'Reilly with another plaque sample showing an unusual...
Look at that. Do you see? It's kind of chasing the bacteria around. How does it know that thing is a bacteria? Because the cell membrane of the bacteria lacks the right markers. Understand? So it recognizes it. But you see how it's changing its shape by changing the cell membrane shape? Does that make sense? It got it. And now it's going to do... anybody knows the name of that? What it just did? This topic? What was that called? What it just did? No? Yes? There's a name for that in cell terms. You're supposed to know. You see? It could have been... Yes? Phagocytosis.
Every second of your life, you are under attack. Billions of...
Phagocytosis. When a cell eats something else. By the way, fag, not F-A-G. Okay? P-H-G means to eat. Okay? Alright, for example, a cannibal is called an entropophagy. No, entropophagy is cannibalism. It's another name for cannibalism. And tropo means man. So it's to eat man. You know what I'm saying? So a man that eats man is engaging in entropophagy. You know what I'm saying? So phage means to eat. Right? Now, what? You want a fag KFC? KFC? That works. I wouldn't say that very often, though. I wouldn't say that very often. Alright, so what is the function of the cell membrane? Alright, okay. Find 4 main components of the cell membrane. Okay, so let's see that. Cell membrane.
If we are what we eat, technically cannibals are the only normal people.
It's not really making
So, okay, the first thing, alright, the majority of what you see here, guys, the majority of what you see here is the red and yellow stuff, right? Okay, what is that? Yes? Phospholipids, Michael. Yes, very good. Get up, Lego. Okay, now, we don't have to just say what it is, we have to say what it does. So the phospholipid is the main component. The main component of the cell membrane. Okay? Now, the cool thing about them is that they have this polar head and these tails which are non-polar. Now remember when we did the models of the molecules with the candy? One of the molecules that we did, it was a large chain of fatty acids. That's basically what this is. This is just fatty acids. And it's non-polar. Alright? Fatty acid chains. The polar head has a bunch of things, including phosphate. Right? Now, phosphate is charged. Now, the thing is about the polar heads is that inside the cell there's water. Outside the cell there's water. So the polar heads are attracted to water. At the same time, remember how water has surface tension? Oh, I gotta show you guys this.
Okay.
Even if it makes me late or my lecture. Water. Oh, hold on a second. Now, what am I trying to show you? It's water, surface, tension, space. You got to see this. Check this out. This is so cool.
The question is, if you get a cloth dripping wet without gravity and you wring it out, what's going to happen? What will happen to a wrung-out cloth? So, and had to use equipment that was here on board the space station. We might have the coolest washcloths ever here on the space station. I'm going to show you. Here's one of our washcloths.
They're dehydrated, so that's kind of great.
And it's packed. It's put down into this little tiny hockey puck so that it saves space. But when you open up a hockey puck and you pull out your washcloth,
He's in the International Space Station. Can't you use it like a headset or something like that? Like seriously.
this is the one I'm going to use for the experiment today.
By the way, totally controlled chaos, isn't it? Like if I work on that place, it'll drive me crazy. I'm like, what do we do with all these cables? You've got to do something about these cables. No, seriously, it's an incredible control chaos. It's actually everything is organized, and yet it looks disorganized because it's a bit. What is the word for it? No, it's a lot of stuff to pack into small space. So it's a bit. They hoard a bit, you know.
And so
I'm skipping this stuff because it's just blah blah blah.
but bags don't hold water in space, so instead I fill the water bag. This has drinking water in it. And I'm going to squirt a bunch of water into this washcloth.
Oh, that's so good. Now you see the cohesion, right? How the water sticks together. Now he's going to absorb by capillary action. See, capillary action doesn't happen in space. Why not? Or does it? It has nothing to do with gravity. It has to do with sticking to things, right? So it should still happen, and it will. It will stick to that thing.
Okay.
So it's a wet cloth.
Soaking wet washcloth. Get the microphone so you can hear me while I'm talking. And now let's start wringing it out. It's really wet.
No, notice how nothing is dripping. Look what happens to it. Look at it. It sticks to it. Adhesion and cohesion.
It's becoming a tube of water.
Look how it goes over his hands. Surface tension. That's really cool. Come on.
The water sort of has it stick to my hand.
Guys, did you guys know that the International Space Station has a TV channel that you can watch, and they do this kind of stuff all the time? So those of you who are science geeks, tune in. It's on YouTube. It's a channel. He was actually, I don't know if you heard in the background, there was clapping. He's actually answering a question from high school students from a school in California. So they were talking to him live, you know, and that would be pretty cool. All right, so anyways, the surface tension of water that you just saw, because water's on the inside, is pushing the cell membrane from both sides. It kind of helps water. See, this is important, guys. Because of the polar heads attraction to water, that surface tension is going to help hold the membrane together. And then the tails love each other, and it'll hold, so then you make two layers of this. And it kind of makes sense. None of the tails have to touch water. Both of the tails touch water inside and outside. Both of the heads are pushed by the surface tension. The cell membrane holds together. And that's really cool. Now, not modern cells, like ancient cells would just be that. But modern cells actually have networks of fibers on the inside. We're going to see this in the video. Now, next class, the class after next class, I'll show you a video, really cool, that shows you the cell membrane, like how it is. By the way, all of these videos which I show you guys are in the videos and web tutorials folder. I would strongly recommend you that from now on, every topic, you visit that folder and look at it. Because I'm never going to have enough time to show you all of them, but they're really good. And every time you see animations in biology, nuclear medical media, or bioflicks, you got to watch those. Those are really, really great. They're short. They're like a minute tops. And it will show you the biology like visually, and you'll make it like, oh, I get it now. So there's a video about the cell membrane by Bioflicks, which is amazing. It will show you how the membrane is. And I will show you next class, but there are more videos which I don't have time to show you. But either way, this phospholipids is the reason the cell membrane does what it does. We'll talk about that in a second. But the fact that it holds together has to do with the polar heads and polar and non-polar tails. All right? The second component, what is this blue stuff here? Let's see. Hold on. Michael? Where's Michael? What is this blue stuff probably? It's labeled by, so you can...
I don't want to know that.
Proteins. Michael, what are proteins made of? You built them 3 classes ago, last week. Building blocks of proteins.
Yes, Michael knows.
Yes, Michael. Nope, not proteins. Nice try though. Starts with an A. Michael. Michael starts with an A. Amino acids. Okay, listen. You grew up in a country of multiple choices. Like seriously. That's not knowing. If you need the options to know on a factual question, trust me, there's a robot coming for your job. Like seriously, there's a robot coming for your job regardless. We're all screwed. But aside from that, you're really out of a job if you can't even memorize a fact. And that's an easy one. And it's a crucial one, an objective. It's on the objectives. You gotta know the objectives. Amino acids. But you knew it. You just need a little hint. But let's not do hints. You gotta know that one. Amino acids. What is it?
Amino acids.
Builds what? Okay. Alright. Okay. So, okay. Proteins do lots of jobs. So we gotta talk about the jobs, okay? Shh, quiet. Guys, all this side talk, please. Give me a job, Frank. That proteins do. Yes, and water is wet. Now I'll give you something else. Yes, Michael. Yes. There are carrier proteins, there are channel proteins, and there are pumps. And these things help move things across. Move things across the membrane. Make sure your notes reflect this information. What else though? There are proteins here that are doing other things. There are proteins acting like anchors. Anchors for the cytoskeleton and the extracellular matrix, which is this stuff here that connects one cell to the other. Alright? There are recognition and communication proteins. These are the ones which allow the cells to receive signals from the outside or to tell these cells from the outside, we belong together. We are part of the body. When you get cancer, one of the first things that happen is that the signals that's supposed to be there become funky, and then your white blood cells, the ones you saw hunting the bacteria, they know that the cancer cells are not supposed to be there and start attacking it. If it wasn't for that, the cancer would spread a lot faster. Does that make sense? So these flags that you see there are markers that tell us how we belong. Have you heard about blood types? That's what that is. It's the flag that you carry. And if you put the wrong blood type in the wrong person, what would I do with it? Fight it. And then what happens? You could have a reaction that causes a problem. So that's why it's so important. But proteins have a lot of jobs in the membrane, as you can see. Okay? These are some of the most important ones. All right? 3. You see this little, by the way, notice how some proteins go across the entire membrane, while others just sit on it. The ones that just sit on it are called peripheral proteins. The other ones are called integral or transmembrane proteins. Now you see this protein has a little tail in there. So these are called glycoproteins. They're basically a protein with a little sugar tail. So so you have this sugar chain or polysaccharide. If you don't know what a polysaccharide, just write sugar chain. All right? So you have this polysaccharide, and then you have a protein. But if you look carefully, there are some of those tails also hanging on the fat, on the actual polar heads. So there's another version that has the same sugar chain, but except it's anchored on the fat. And those are called Glycol Lipids. So this is the third component. Glycoproteins or glycolipids. You see them here. This is a glycolipid because it's attached to a fat. That's a glycoprotein because it's attached to a protein. But either way, those carb tails are involved with recognition and communication as well. Alright? So let me show you what they are for. And last but not least, the fourth major component, what have we talked about? We already talked about the little tails. We talked about the proteins, the red and the yellow. What's missing? These guys. Anybody knows what that is? Yes, Gabby. Cholesterol. Now, cholesterol is a thickening agent on the membrane. So, what does that do then? What does it sound like it does? It makes the membrane what? Well, literally more kind of gooey and sticky. Now, we're going to talk about this in a second, but this whole thing, the membrane, is a fluid. You see these little red blobs here? They're like floating around. Not next class, but the class after next class, I'll show you a video of this. And you're going to see that the membrane is just kind of fluid. All right? Hell, it helps me get the message across. So, if you look,
flavor with America's no compromise pizza.
no comment. Save it. Save it. All right.
Between the living machinery of the inner cell and the harsh conditions of the outside world stands the cell's plasma membrane. As crucial as this barrier is, it's surprisingly flexible. Push it and watch it move. Poke hard enough and it might break and begin to regroup. The lipid molecules of the membrane naturally assemble in a double layer because their tails repel water as their heads attract it.
A little older. I have that one in my videos. By the way, all this stuff I found on YouTube and I collected already in that folder.
Between the living machinery of the inner cell.
That video. I'm going to find a new one. I think that's the same one I just watched.
Between the living machinery of the inner, these molecules are not stationary.
Either way, I have a better one that will show you that later. It's a bioflix cell membrane. I'll show you that to that class. But the idea is the whole thing is fluid. Each of these little fat things are flowing like a river of fat. Right? Ever seen oil bubbles floating around the water? Yeah. Kind of like that. But ever so often, there's a bit of a raft or an island. And those islands are basically made, stop the side talk. They're basically made by cholesterol kind of grouping, right? Now, if I live in the desert where it's really hot, and it makes it more likely for my cells to become fluid, I need more cholesterol to keep my membranes from becoming too fluid. If I live in a cold, less cholesterol is bad or good. If for some reason cholesterol stops being in the membranes and becomes more part of the blood, now you increase cholesterol in the blood, the thickening agent thickens the blood instead. And it makes it more likely for you to get clots, right? That could cause heart attack. All right? But you do want cholesterol just, in the right place. And the membrane uses it to thicken it when necessary. Okay, so that's that question. We gotta keep going. Pick up speed a little bit. All right. Now, why is the membrane semi-permeable? Semi means almost. Permeable means what? What does that sound like? Less things through. So almost less. Okay, so the idea here is that there are things allowed and things which are blocked. So this is called semi-permeable. It blocks certain things but allows others. But they want to know what makes it that way. What makes it that way is the double layer of fat. Alright? Because if you look at the thing, what is the majority of the layer made of? The polar heads or the lipid tails? Look at it. The lipid tails take up more space, doesn't they? So now these things are non-polar. And these things are polar. Now, that means that anything that's polar is going to have to get through the big volume of non-polar things here. And since they don't like each other, they're going to repel each other. That means that things which are polar, or things which are charged, or things which are too large, cannot get through the membrane very easily. Although there are some viruses that they put a fat layer around them. And that, since they're surrounded in fat, they go through the membrane, no problem. So, things which are allowed are things which are non-polar, like fat. Or very small, tiny things can possibly get through. So what are some examples of these things? That's the next question. Examples of things which are blocked versus non-blocked by the cell membrane. So, for example, carbon dioxide, oxygen, and fats, such as, for example, steroids, hormones, get through with no problem, or cholesterol. It's like the membrane is not even there. I ever heard about this thing that David Copperfield did, that he walked through, he walked through the Great Wall of China, supposedly. Right? It's like you just walk through the wall and it's not even there. That's what things which are fat can do. They can literally walk through the cell membrane if it's not even there. Right? The membrane is not there. It acts as if the membrane is not even there. Because it's non-polar. However, a lot of other things which the cell needs, both to get rid of and needs to survive, for example, waste products, right? Salts, which cells want to get or get rid of, water, which the cell might need to get in or get rid of, amino acids, which are the building blocks to make protein, which is the whole point of a cell. It's a protein factory. So it needs the building blocks to do that. Sugars, the cells need to power their chemical reactions, right? The energy. Proteins, which is usually what cells make, they need to export. Or they need to import, to destroy, to get the amino acids to make new proteins. Right? All of these things, which are things the cell, or bacteria, well, the cell doesn't want that, but that's a good thing probably. Viruses, cell doesn't want that either, but it's fine. But a lot of these things are things the cell wants to get in, but it's blocked because they are large, charged, or polar, all of these things. And so, the cell membrane is semi-permeable, allows certain things in and out, no problem. It's probably a good thing that these guys get in and out, no problem. So as long as you have a lot of oxygen outside, oxygen will tend to go inside, no problem. Right? Carbon dioxide is produced by the cell. And since it goes to the membrane with no problem, it just leaves the cell with no problem. Right? So it's probably a good thing. Steroids can be fast messengers because they get through the membrane and it's not even there. So the message gets through very quickly. But what about sugar, amino acids, water, salts, getting rid of waste? How does a cell do that if it blocks it? We'll talk about that in a second. All right? Now, what is the difference between the cell? Before we talk about that, a little break. There's cell membranes and then there's cell wall. All right? Now, if you look at the cell membrane, if this room was a cell, the cell membrane would be the size, imagine a pencil. Draw a line with that pencil on the floor. The thickness of that line would be the size of the cell membrane if the cells were the size of this room. So it's a very thick. It's a thin little layer. Very thin little layer, right? And so that picture that we saw was would be very, very magnified, right? Of course, there's nothing that can magnify it that much. Well, actually there is, but nothing that we can easily use to magnify it that much, right? So there's no real picture of a cell membrane up close, like there's only drawings, right? But the idea is, though, that small little layer does all that job. But the cell wall can be very thick. The cell wall can be thousands of times thicker than the cell membrane. But people often hear the word wall, they think, oh, it's for protection, right? And it is, and it isn't. All right, so let's think about, so remember to answer this question, what does the cell wall do? The cell wall does act somewhat for protection against parasite. So it's parasite protection or toxin protection. For example, if you try to destroy a bacteria that have a very thick cell wall with an antibiotic, the antibiotic might not get through and never kill the bacteria. Some bacteria are resistant to antibiotic because they have special cell walls that make them resistant to it. Does that make sense? Plants, they have parasites that live in there just like you and me. Because they have such thick cell walls, it makes it harder for the parasites to get through. However, that's not the primary role of the cell wall. The actual role of the cell wall is for structure. So I'll give you an example with the plant cells. Okay? So the plant cell looks like most of them will look like a little box. Not all of them, but most of them will look like a little box. And normally the cell membrane is just at the wall like this. So you can't even tell that the wall is there because the membrane. By the way, we're going to do a lab. It's going to on Friday. We're going to do something that's going to let you guys see this. Right? You can try it, doing it. If it doesn't work for you guys, I'll at least show you a video of it. But you can always, if for some reason on Friday if you run out of time and you can't do it well, you can always do it on your own time. It's easy. We get the LODS cells from the water. We put salt on them, and you can see the cell membrane do this. Because this is the thing. If you put a lot of salt outside, the cell tends to lose water, right? Because water tends to go to where there's more salt. When it does that, as the plant cell loses water, that cell membrane kind of shrivels inwards, right? So then instead of being at the wall, the membrane shrivels, and you can see it. You see the membrane, but the wall stays where it is. Now, imagine this cell is part of a large bark of a large tree. Are you with me? What would happen if it wasn't for the wall when the cell lost water and the membrane collapsed? What would happen to the bark of the tree? Think about it. The whole tree would literally what? Shrivel. Imagine that happened to a tree. Does that sound like a good thing for a tree? No. So, on the other hand, if the box, the cell wall, maintains the structure even when the cell loses water, the tree's not just going to fall apart. Not to mention that the barks of the trees are actually, the majority of it is dead wood. That's just the cell walls. That's all that's left. Cellulose. Thick cellulose walls. And that is really what supports the plant. Are you with me? So the cell walls are, yes, you're not going to say the bark doesn't protect the tree. You're not going to say the cell wall doesn't protect the cell. However, the primary role is maintaining the structure of the cell and helping maintaining the pressure of the cell. Does that make sense? Moving on. Why is homeostasis important for life? What is homeostasis again? Hold on, Margie. To keep balance, equilibrium, all right? Give an example, Gabriella, of your body maintaining homeostasis. When you sweat because it's too hot, your body's trying to keep it cool. It's called a negative feedback loop. Something is off balance, you do the opposite to try to bring it down, right? Give me another example, someone. Yes? Shivering when you're cold. What do you do when there's less air in the room? Do you know what happens with your body? Yes? Why, but if there's little air in the room, what happens to you? Do you know? Yes? Yes? You actually start getting sleepy. Why do you think your body gets sleepy? Yes? So you literally consume less oxygen. Does that make sense? So it's all types of adaptation. Now, likewise, at the cellular level, your cell must maintain balance. And the cell membrane, of course, is very important with that. So let's talk about some ways the cell membrane does that. And let's go back to those concepts over here. First of all, we have to contrast active versus passive transport. All right, so I'm going to erase the cell wall bit here and talk about active versus passive transport. Remember, guys, this is a review. If you don't study at home, this is gibberish. You got to study. Otherwise, if this is the first time you're being exposed to this, it's not enough for you to learn. You need to take notes, see visuals. All right, watch Bozeman, Amoeba Sisters, or even my videos, whatever you want. I have visuals. This is a review. If this is the only learning you're doing, God help you. Well, I don't know if that's the right expression. But anyways, passive versus active. Okay? Passive. Okay? Passive. Passive transport is when you go from high to low concentration. Now, if you open the gate of a dam, do you need to push the water, or the water is just going to go because of the potential? See, it's based on the actual potential and kinetic energy of the molecules. You do not need to do anything. The molecules just change to do that. I said in the video, if we're all blinded molecules moving crazily and bouncing from each other, and you open this wall, a door, since there's no one outside, there's zero chance of one of us leaving. But if we're all chaotically bouncing, there's going to be a chance of one of us leaving. But as more and more of us leave, eventually there's an equal chance of people leaving and coming in. That's diffusion. It's a natural process. No one needs to spend energy. On the other hand, if you're trying to send the water back into the dam after it flew down the river, you're going to pump it up there. Because that's putting it from low concentration to high concentration, and that requires energy, which in the cell comes in the form of ATP, a molecule that carries that energy. So you need to spend energy to do it. All right. Now there's another question here. Simple versus facilitated diffusion. So this side here, the passive side, remember we call that side diffusion. Natural movement of things from where there's a lot of them or when they're highly concentrated. By the way, not from there's a lot to where there's a low. It's from the concentration is high to where the concentration is low. So it's not that there's a lot of water behind the dam. It's that there's more water behind the dam than there is here. Remember the cup thing that we did? It doesn't work if there's more water on the cup and you try to make the water go backwards. Are you with me? It won't do that because it tends to go from where there's a lot to where there's less. Now, having said that, there's two types. Okay? There is simple diffusion and facilitated diffusion. Facilitated diffusion, these things right here, okay, need to go through facilitated diffusion. While those things right there need to go through simple. Basically, the membrane is a selective barrier, right? Selective barrier. So anything like that just gets through it like it's not even there. But things like this need a canal or a surface protein, something that's going to allow it to carry it through. The cool thing about canals, though, is that you can open and close it. You can close a part of a canal if you want. You can close a bridge if you want, right? So this allows the cell membrane to control whether or not those things come in. Does that make sense? Meanwhile, you also have pumps. We talked about pumps, which if you want them to go out, even though they don't want to go out because there's more of them already out. For example, let's say there's a lot of waste out, but the cell's still trying to pump the waste out. Well, you can still do it, but it costs energy to do it. Let's say, for example, there's a waste product. If I just open the gate, the waste will try to go in rather than go out if there's more waste out than in, based on this principle. Correct? So I close the gate and pump it out. Does that make sense? When do I open the gate? When I want things to go in naturally. Are you with me? Does that make sense? Right? So that's what the cell does to control what it does, and I talk more about that in the video. Now, the last of this, let me see, 10 minutes left? Okay, we're going to start then. The rest of this is talking about examples of actual things moving in the body and how things get through membranes in the body. So these are the options that we have. Okay, by the way, before I continue the options, let's talk about endocytosis and exocytosis. So this is endocytosis here. Images, perfect. I don't have to do much. Okay, so, whoa, that was a good one. I want that one. Real image, make it smaller. Okay, when something is trying to get in the cell, and the cell literally projects these little arms, like we saw that cell eating it earlier, and swallowing the whole thing whole, that's called phagocytosis. You see the cell projecting the little arms and eating it. We saw this in the video, right? On the other hand, if the cell just pinches inwards and grabs whatever's in there, it's called peno. Peno means drinking instead of eating, right? So penocytosis means the cell is just taking a sip. You follow what I'm trying to say? From what's outside. And a lot of things get inside like that. Now, This one is just like, I want to take a sip, but I only want certain things to get in. There's a lot of things out there, but I only want that in. So once that connects in a membrane protein, then it sips. So that's to make sure that only what it wants goes in. So that's called receptor-mediated endocytosis. Exocytosis is the opposite of these things. Exocytosis is when, instead of coming in, look, there's a vesicle, it just kind of merges with the cell membrane and throws the things out. Does that make sense? Now, that's to move large things, enormous things. Okay? All right, so now back to the questions. We're going to go to the blackboard. I'll do it. Yeah, go for the blackboard. Can you move that towards the blackboard, please? Carefully and slowly. Yes. The camera is temperamental. Oh, it already did. It's too late. Temperamental little camera.
That it's temperament. Temper? There or where?
Yeah, closer.
Where did you
All right, so remember, we're not going to finish this today. Don't forget to bring your stuff next class. The class after next class, we're gonna finish this. We're gonna do the microscope activity and whatever else we have time to do on that day. Okay? Now, for each of these things, we have to talk about what process. By the way, we're going to continue this next time. So we're going to talk about what process lets things through. All right? And then we're going to talk about examples in our body of this happening. All right? So for each of these things, we're going to have to see, is it passive or is it active transport? Is it small transport or is it bulk transport? All right? Does it require ATP or not? Right? And is it assisted or not? And then an actual example in biology. So that's what we're going to try to do for each of these things. And now, of course, I don't know if they're doing announcements today or not, but if they do, we'll have to go over them. All right? So then, again, all right, so then look at that. Hydrogen ions move down the concentration gradient. So hydrogen ions is a charged thing. According to our rules, does the membrane like charged things to go through? No. Now, remember, whenever I go from high to low, does that mean energy or is that normal? Water down, high to low. Is that normal? Or is it, that's a normal way of things. Okay? So, let's do our rules. Would that be passive or active then, moving down the concentration gradient? Passive. Okay, so hydrogen ions down, concentration gradient. So that would be passive. Okay, but let's think about it. Is that going to be, they're small little things. So it's not a bulk transport, that's a small transport. All right? Now, this thing is going to go naturally down the concentration gradient, so it's simple diffusion. All right, so it's just our diffusion. It's just diffusion, so that means no ATP. No ATP. All right. Now, actually, I'm going to do this like a chart. Makes more sense. Remember, we're not going to finish it today, but it's going to be easy as a chart. Passive versus active. All right. Now remember that passive requires no ATP, and active requires ATP. Okay. This is just a name for the type of transport that we're talking about, and then the options: is it bulk or is it small transport? Who assists? If assisted, who assists? And then the example. All right, and then we do the same thing. Hydrogen ions down the concentration gradient. If it's going down, that's a natural thing. Therefore, it's passive. It does not require ATP. Hydrogen ions are tiny, small. But because they are charged, the membrane blocks them. So the only way they're going to get through is with help. So they're going to use a channel to get through. There's got to be like a canal to get them through that membrane. Right? So this is called facilitated diffusion. An example of this is in photosynthesis and cell respiration. There's a process called the ETC. There's a process called the ETC on both cell respiration and photosynthesis, where this is going to be very, very important. And in fact, the cell uses this to make energy. We'll explain more of that with cell respiration. Okay, water. Water, which is, by the way, the movement of water is called osmosis. All right? But water moving down the concentration gradient. So it means from where there's a lot of water concentration to where there's low water concentration. That's the natural way of going. So that's going to be passive. Water is small, no problem. But water is polar. So it's going to need a channel. It's going to need a channel. So the example of the water channel is aquaporin. Aquaporins are little channels that allow water through the membrane. Tiny little holes in the membrane to let water in and out. Because it's water, we call it osmosis. All right? Which is a type of facilitated diffusion. Because it's using those aquaporins. Stop talking over me. Right? An example of water moving through this is in your kidneys. Right? In your kidneys, when there's too much water in your body, the kidneys absorb the water, and that's why you pee. Right? So your kidneys are actually regulating homeostasis, the amount of volume of your blood. If it starts becoming too much water in your blood for some reason, the kidneys take that water out by osmosis. Does that make sense? All right? The next one, it says oxygen down the concentration gradient. So again, oxygen moving down. Okay, down means passive, from high concentration to low concentration. It is small transport. Now the thing is that oxygen is nonpolar. So it goes through without help. No help. Which means it's just simple diffusion. It's like the membrane is not even there. Oxygen just leaves or comes in as it wants. An example of that is literally between your lungs and your blood. So in your lungs, there's these little tiny air sacs called alveoli. All right, inside the lungs. And they're full of oxygen. Yes. Meanwhile, there's your blood. Your blood comes in depleted without oxygen gas. Right? So then what happens? The oxygen just jumps. Does that make sense? To the blood. So the example of this is in breathing. Breathing. When you get your cells, is the opposite. Your red blood cells are full of oxygen, and your cells are depleted of oxygen. So then the cells give the oxygen to the thing. You know what I'm saying? So as long as your blood comes in without oxygen, right, your lungs keep giving it oxygen. As long as your cells need oxygen, the blood keeps giving oxygen to them. So this is a method, natural process of exchange of materials. Very important. Carbon dioxide down. The same thing is true here. Passive, small, no, simple diffusion, and the same thing backwards. When you get to the blood comes in full of carbon dioxide from the cells that dumped into it, right? The red blood cells come full of carbon dioxide. And then the lungs are low in carbon dioxide because they just put it out. Oxygen in, carbon dioxide out, right? So that means the carbon dioxide tends to go to the lungs. Right? Now think about this though. What happens if your blood becomes rich in carbon dioxide? So you hold your breath for 2 minutes. The more I hold my breath, the more my cells are breathing, my carbon dioxide levels in my blood are increasing. It gets to a point that the blood is rich in carbon dioxide. Right? Perhaps even more than inside a cell. So that means the carbon dioxide actually starts going inside the cell. And that actually hurts the cells, because the cells want what? Carbon dioxide? Out. Which just means if you stop breathing, that's why you die. You die literally of carbon dioxide toxicity. Does that make sense? Right? Because in order for the system to keep working, I gotta make my blood get rid of carbon dioxide at my lungs. You see? But I need no energy for this. This just happens naturally, so long as the system keeps going. And that's the bell, guys. Get out of here. Hey, remind me where we stop off next time. We talked about carbon dioxide is the last one we did. I'll see you next time.